Succession in a jar. Introduction. In this lab, students will be observing changes in 3 pond water environments and counting each type of organism they discover. Students should conclude whether or not the changes in the 3 environments represent succession. Students will need to examine 3 jars, each containing different material. They will then need to prepare wet mounts from each jar and observe their findings under a microscope. In order to do this, students will need to know how to prepare a wet mount slide and practice safe microscope techniques. Preparation and tips. For this lab, you will need pond water containing plant material. You may seed the pond water with protest cultures from a biological supply house, if necessary. In place of cooked white rice, you may also use cooked wheat or pea seeds. However, do not use instant rice. Sterilized spring water may be replaced with boiled and cooled tap water. If class time is limited, you may wish to examine the jars once a week instead of every 3 days. Safety. Wear your apron in the lab and always wear goggles. Remind students to wash their hands thoroughly each time they examine the jars. Handle all slides with care. Lab techniques. Proper instruction should be enforced when allowing students to use a microscope. Always carry the microscope by holding the arm with one hand and supporting the base with the other hand. Look through the eyepiece. Adjust the diaphragm so that light comes through the opening in the stage. Place a slide on the stage so that the specimen is in the field of view. Hold it firmly in place by using the stage clips. Always focus first with the coarse adjustment and the low-power objective lens. Once the object is in focus on low power, turn the nosepiece until the high-power objective is in place. Then use only the fine adjustment to focus with this lens. When finished, make sure students store microscopes with the low-power objective in place. When students are ready to prepare a wet mount slide, make sure they understand the correct techniques. Carefully place the item you want to view in the center of a clean glass slide. Make sure the sample is thin enough for light to pass through. Use a dropper to place 1 or 2 drops of water directly on the sample. Hold a clean cover slip by the edges and place it at one edge of the drop of water. Slowly lower the cover slip onto the drop of water until it lies flat. During the wet mount procedure, an excess of water or air bubbles may appear. Tell students to touch the edge of a paper towel to the edge of the cover slip to draw off extra water and force air out. As with excess water on a wet mount slide, students may need to instead add more water to a slide. To do this, touch the dropper near the edge of the cover slip and release 1 drop of water. Never take the cover slip off the slide to add or remove water. Once the lab is completed, cultures may be placed in a large vessel and transported to the nearest pond to be deposited back into their natural environment. Troubleshooting. Organisms can be slowed down by using ProtoSlow, available from most supply houses. If students are having trouble determining whether or not a jar shows turbidity, ask them to hold a newspaper clipping against the jar. If they cannot read the print through the jar, turbidity has set in. As weeks pass during the experiment, water may evaporate from the jars. Additional pasteurized water should be added to replace the loss that may occur. Results. Jar A should stay clear. No turbidity or living organisms should exist. Jar B should show turbidity, but no autotrophic or heterotrophic organisms. Jar C should show an increase in cloudiness and then decrease somewhat. Autotrophs should increase in number while bacteria decreases. Coppidium numbers rise, then fall within 20 days. Euplotes numbers rise, then fall during days 40 to 60. And paramecium steadily rises. Overall, turbidity indicates the presence of living organisms. Applying lab techniques. In order for students to fully understand the role of succession in an environment, it is important for them to grasp the idea of specific variables playing a role in the changing populations. Certain variables can speed the rate of succession more so than others. Have students collect 2 jars of pond water and observe the various organisms under low power. Then keep 1 jar in sunlight and place the other in a dark cabinet. Observe the changes that occur to each jar. Does absence of light affect the number of organisms in the jar?